Welcome back, everyone, to the next section of the symposium on the mechanisms of cystogenesis. I myself is Balaji Karthik Subramanian, an assistant professor at Harvard Medical School, and Amanda Clark, a postdoctoral fellow from University of Alabama, will be the moderators for the sessions. And our first speaker for the session is Dr. Lloyd Cantley. Dr. Cantley is the C.N.H. Long Professor of Medicine and Professor of Cellular and Molecular Physiology at the Yale School of Medicine. The primary focus of Dr. Cantley lab is to determine the mechanisms of renal tubule development, homeostasis, and repair. And the title of his talk is Physiologic and Pathologic Tubule Remodeling. Professor, please proceed for your presentation. Thank you.
Yeah, it warned me about this before, and I tried to question this, but anyhow. Okay, so these are my disclosures. I realize I'm already way behind. I apologize.
No, Lloyd, don't worry. We just started the timer. We waited, so just.
Sorry. It was clearly weird, and I was trying to figure out why before the meeting started. So if there was something wrong, I had to restart Zoom. Okay, so as mentioned, thank you so much for the introduction, and I really appreciate the opportunity to talk at this meeting. It's a great meeting so far. My lab studies tubule development and tubule morphology and tubule repair. And within that context, we think a lot about PKD, although it's not the only thing we think about. And in thinking about the kidney, I trained at the BI under Frank Epstein. And I remember thinking about the fact that the kidney has your full number of nephrons when you're born. And yet the single nephron GFR increases markedly as you get older. You start out with about 4.5 mLs a minute of GFR at birth and end up somewhere around 110 to 120 at full adult size. So you have about a 25-fold increase in single nephron GFR. And yet we know that the kidney precisely maintains the reabsorption of that filtrate plus or minus 0.1% or something at roughly 99% of most of what you filter you reabsorb. And so that clearly requires a massive morphologic adaptation to deal with a 25-fold increase in single nephron GFR. So back in the 80s, when Frank and many other titans of kidney physiology were doing their work, the term glomerular tubular balance was described from a series of studies that showed that very acutely, within minutes to a few hours, the tubular cells will increase synthesis of and a trafficking of transporters to the membrane at the brush border or not in order to respond to acute changes in GFR. But we were more interested in what happens with chronic changes in GFR, this kind of 25-fold increase. You're not going to be able to upregulate enough transporters to handle that. And so this led us to this term glomerular tubular synchronization as we thought about how do tubules adapt morphologically to sustain changes in GFR. And my thinking about this really started to crystallize when we were doing work on PKD and macrophages, which I'll come to in a minute. And to try to understand that more carefully, we had started using this technology of optical clearing to study a specific site in a tubule. So we can track tubules. This is one Z-stack image that you're seeing to the left here. And we would take 500 micron thick sections of mouse kidney and optically section them at 1 micron increments. So you get 500 Z-stacked images from one section of tissue that would allow you to track tubules throughout the entire nephron from the glomerulus all the way to the collecting duct. And by doing that, we could define a specific site in the tubule that we could then analyze very precisely. So that rather than looking at cyst index in PKD, we wanted to look at what exactly is happening in the proximal tubule or other tubule segments. We highlighted proximal tubule because it was the most obvious that you could pick a glomerulus as the one shown here and then track that glomerulus to a specific point where you got a beautiful, perfect cross-section. And we could do that across many glomeruli and many kidneys and analyze cross-sections of tubules at roughly the same distance from the glomerulus, plus or minus 20 microns. These are mostly about 100 microns from the glomerulus. And you could measure the area of the tubule, the area of the cell body that makes up that tubule, and the number of cells that make up that epithelial area. And you can also actually analyze brush border. You can see the brush border here and lumen. And so that analysis led us to some things I'm going to show you in a minute, but that led me to think about this idea of exactly how does the tubule synchronize the morphology of the tubule to the imposed GFR. And so the first model we looked at to ask this question was a model of just aging of a mouse. So a 4-week-old mouse is pretty young, has small kidneys, 18-week-old mouse, bigger kidneys, and with that, a bigger GFR, just like humans. And we did this same type of imaging, and you can see here's an average cross-section of a 4-week-old mouse and an 18-week-old mouse. And each dot's a separate tubule across, I don't know, maybe 5 mice, if I remember correctly. And you can see that their tubules are generally smaller in a 4-week-old mouse, bigger in an 18. This is obvious. It makes sense. Epithelial area smaller, gets bigger as the mouse gets larger. And the tubular area, very small to begin with, and also gets larger. But look at the difference in scale. So here we're at roughly 25 square microns versus about 1,000 to 1,200 square microns for the tubule. But if you want to look at this more specifically, you analyze each individual tubule. So as opposed to just looking at dot plots like this, you're looking at this bar represents one tubule's area, and the dot on the top represents the epithelial area for that same tubule. And that's very similar for each individual tubule. There are big tubules and little tubules in a 4-week-old mouse, but the epithelial area is always very tightly matched to that particular tubular area, and the same for an 18-week-old mouse. So that ratio is extremely linear with a very tight R value. Even though there are some tubules, for example, an 18-week-old mouse that are smaller than those in a 4-week-old mouse. So this maintenance is very tight. And it turns out that the same thing is true in hyperfiltration. And we looked at that by doing nephrectomy at 6 weeks of age. And we picked this age because it'll be relevant when we come to the knockout data, where we do nephrectomy of one kidney and then wait 2 weeks. And then 8 weeks, we analyze the remaining kidney. And that remaining kidney got bigger. As you can see, this is the sham mouse, which still has 2 kidneys. And if you compare the left kidney of the sham mouse to the remaining left kidney and the nephrectomized mouse, you can see that it got bigger. The single nephron GFR must get bigger because the GFR per kidney gets substantially bigger. This is work from Tong Wang, who's a collaborator with us on this. And then the tubular area gets bigger again, the epithelial area gets bigger, and the lumen area gets bigger. All of these changes are very similar. And here it is at the individual tubule level. So you can see each bar is an individual tubule. Again, the dot at the top is the epithelial area for that tubule. And there's that very tight linear relationship. So overall, the tubules get bigger than they were. Each individual tubule may be slightly smaller or bigger than a tubule in a non-nephrectomized mouse, but the overall area is larger. But again, with this very tightly synchronized morphology, which presumably is optimal for the reabsorption of that increased GFR. And this is just a composite picture showing the young mice, the 8-week sham operated, the 8-week nephrectomized, and the 18-week mice, showing that there's a spectrum across those of all these different tubules, from the smallest to the largest, but that regardless of the particular condition, once a tubule has established a particular size, the cell body, here we analyze not just epithelial area, but we broke it down into cell body and brush border. So the full circles are cell body and the open circles are brush border. You add those together to get the epithelial area that would be way up here, as I showed before. And down here, the stars are the lumen. And you can see that ratio is maintained for the cell body to tubular area, the brush border to tubular area, very tightly controlled, and the lumen to tubular area, very tightly controlled. So the kidney morphologically keeps this the way it wants it. And our hypothesis is that the cilia are the sensor that detects the sustained change in GFR in some way that I'm not going to get into today. We're doing work with IFT88 knockout and trying to analyze this part of it. That data is not complete enough for me to present it yet, but that the polycystins act, this is the Steve Somlo model, the cilia-dependent cyst activating signal coming from the cilia, and the polycystins are the brakes, except from my perspective, it's the cilia-dependent kidney tubule synchronization signal coming from the cilia normally with the polycystins as the brake. And so to see if that played out pathologically as opposed to just physiologically, we asked the same question in the PKD model of mice. And this is the Pax8 Cre, doxycycline inducible Cre, Pax8, PKD1 flox mouse. So if you give this mouse doxycycline, you induce the Cre and Pax8 expressing cells, which are predominantly in the renal tubule, and you then knock out whatever's floxed, in this case, PKD1. So we induce the mice from 4 weeks to 6 weeks of age. Then we do that nephrectomy or sham surgery at 6 weeks, and again, analyze at 8 weeks. So this part's the same as I showed before, but they will or will not be induced. If they're not induced, we call them uninduced or UI. So those should be the same as wild type. And then the PKD1 knockout in this case were induced. And you can see that in the sham operated mice, very similar to what I just showed you in wild type mice, in the sham operated mice, they have PKD1 knockout, and you do see some cysts already at 8 weeks of age and some dilated tubules, but also tubules that are pretty normal. Whereas in the mouse that's nephrectomized, that remaining kidney, you can see almost all of the tubules are very dilated and much more cystic. And that plays out over here with the cyst index, where the uninduced mice, this is what I was going to say, as I showed you before, have a very small area that would be considered cystic. Nephrectomy, that tubule enlargement that I showed you with nephrectomy in a control mouse, results in an increase in lumen area and therefore a higher cyst index if you just do this as cyst index, although these aren't cystic at all. The sham mouse is the typical PKD knockout. At 8 weeks, it already has a detectable increase in cyst index, but nephrectomy really increased that. So now you come to the individual tubule analysis, and you can see this is what a sham tubule looks like. This is an uninduced nephrectomized kidney. So these two would be just like wild type that I showed before. This is the PKD knockout sham and PKD knockout nephrectomy. And the little bit of a surprise was that, again, the sham operated mouse PKD has a bigger tubular area, a bigger epithelial area, and a bigger lumen area than control. Nephrectomy further increased that tubular area, but the epithelial area really doesn't change much. And the big difference is that the lumen area dramatically increases with increased filtration. So you can see it's up here close to almost to 1,000 square microns in the lumen area. And if you look at it, you can see that the composite epithelial area here isn't that different than this tubule. It's the lumen that's so much different. And so this thing comes back to the data that actually triggered a lot of these thoughts, which is what are the effectors of this pathologic tubule remodeling in PKD? Why is it the tubule gets so much bigger? And we had shown a few years ago, more than a few years ago now, that macrophages surround these cysts. And it doesn't matter which model you use. You can have a PKD knockout with PKHD1 Cre, the PAX8 Cre, the PKD2 WS25 mouse. In all the models, macrophages infiltrate quite extensively around the cysts. And so we had already published that if we knocked out the homing signal for macrophage recruitment, which is MCP1, macrophage chemoattractant protein 1, at the same time that we knocked out PKD1 in tubules, we could suppress that macrophage recruitment. So here's the genotype of the mouse. It's that same PAX8 regulated Cre, which is doxycycline inducible, with both PKD1 now and MCP1 fluxed. And we induced from 4 to 6 weeks again, same model, but now we analyzed the kidneys at 8, 12, and 18 weeks. No nephrectomy here. This was the previous data. And you can see MCP1 is highly upregulated. If you knock out PKD1, that's these black bars. So here's MCP1. Normally it's very low level. If you knock out PKD1 at 6 weeks, it's already starting up a touch, but at 8 weeks, markedly increased MCP1. And with that, a big increase in the number of macrophages. If you knock out that MCP1 in the tubular cells, you can see you have a substantial reduction. So most of the MCP1 here is coming from the tubular cell, not all of it, but most of it. And with that, a substantial reduction in the macrophages. And with that, a decrease in the kidney size, kidney weight to body weight ratio, and better survival of the mice. So clearly those macrophages coming in are accelerating the cyst growth and doing something bad. We had, in that original paper that we published, we had described cysts that were already fully developed, and we were analyzing those kidneys at fairly late stages of those cysts, and had found that the macrophages that we detected in those established cysts were alternatively activated. But the, again, somewhat surprise in this data was that when we looked at this 8-week time point where we're already seeing the increase in cyst index that I showed you earlier, that all the macrophages being recruited at that early time point were pro-inflammatory macrophages, expressed high levels of IL-12, TNF-alpha, INOS, and really no alternative markers. And that this pro-inflammatory activation correlated with injury to the tubules, identified here by Chem1 and by those images I showed you. So now we ask in this new work that's unpublished still, what happens with nephrectomy to MCP1 and macrophage recruitment? Are they playing a role in the physiologic adaptation to increase infiltration? And so in the uninduced mice, so no knockout of anything, when we do nephrectomy, again, we now see a slight increase in baseline MCP1, not impressive, but definitely statistically significant. And with that, an increase in the number of macrophages, but not pro-inflammatory macrophages, no NOS2 expression, no TNF-alpha expression. So macrophages do appear to be part of the physiologic response to increase infiltration, but they're not pro-inflammatory. Whereas if you do this in the PKD knockout, so SKO here stands for PKD knockout, single knockout of PKD. Now you see that PKD knockout by itself increased MCP1. I already showed you that. But if you do nephrectomy, a much greater increase in MCP1, a much greater increase in macrophages overall with nephrectomy in the PKD1 knockout, and very inflammatory, high levels of NOS2, TNF-alpha, not alternatively activated at that early 8-week time point. So the same phenomenon that we see just with PKD knockout, but further exaggerated. And so what are those pro-inflammatory macrophages doing? Why is this impacting the tubule morphology? And that goes back to that original paper where we, or the paper where we knocked out MCP1, where when we looked at the tubule morphology of the PKD1 knockout, we saw this subset of tubules that I'm showing over here on this side, in which the tubular area is huge, as big as it gets, basically, but the epithelial area was quite small in those tubules. And this is what they look like. They were basically what we would call cysts, right? Very enlarged tubule, very large lumen, and flattened epithelial cells. And if we knocked out the macrophages, that went away. You didn't see that phenotype at all. So somehow those early pro-inflammatory macrophages were driving this. All this data is at 8 weeks of age. And when you look specifically, so we had always assumed that this was going to be related to proliferation. But when we looked at these tubules, we found that, yes, they have a very large tubular area compared to the epithelial area, shown here and quantified here. But there was no increase in the nuclei. I remember I showed you, we could literally measure the number of nuclei in each of these cross sections. So this dilation was not proliferation dependent. And so, I throw this in just to assuage everyone's concerns. There is definitely proliferation when you knock out PKD1. There is this autonomous effect that you can see even in the absence of macrophages. So that would be the dotted line. So compared to a control mouse, there's always more proliferation of the tubular cells in a PKD knockout at all the time points. But after 8 weeks, there's this macrophage-dependent further increase in tubular cell proliferation. And that correlates with the upregulation of arginase 1 at this latter time point. Remember, here at 8 weeks, you really don't see arginase 1 expression. But by 12 weeks, they're switching from pro-inflammatory to alternative, and there's a big proliferative component. Either way, the tubules are getting big, and something is doing that. And so that led to the last part I'm going to talk about, which is how is the remodeling of the basement membrane actually regulated? And to answer that, we turned to RNA-seq. So we did bulk RNA-seq and looked for all of the different metalloproteases that might regulate the tubular basement membrane. And we identified this one, ADAMTS1, that we confirmed by PCR. Here's baseline in a control kidney. Here's a PKD knockout, much higher expressed. It turns out to be the most increased in abundance and in significance of all the metalloproteases. Each of these dots is a metalloprotease. And when you look at single-cell RNA-seq data, it's mostly made by the proximal tubule, confirmed by FACS analysis here. And so what is ADAMTS1? The name stands for a disintegrin, a metalloproteinase with thrombospondin motif 1. I'm sure that excites you. It didn't me. But when I read about it, I got interested because it cleaves Versacan, which is a chondroitin
2 minutes.
Yeah, thank you. Chondroitin sulfate proteoglycan in the basement membrane. And the whole body knockout is infertile because the failure to cleave Versacan prevents follicle rupture. So the follicle can't get bigger if you don't cleave Versacan. And it also was interesting that ADAMTS1 has been implicated in aortic aneurysm formation. If it's excessively active, it cleaves too much Versacan and weakens the wall. So this kind of made me think about polycystic kidney disease. And it cleaves in these two isoforms of Versacan, the V0 and V1. And so we looked for the expression level of ADAMTS1 under these different conditions. And sure enough, if you just do nephrectomy, it's upregulated. So it's physiologically regulated by in the tubular cells by the increase in GFR, but it's even more upregulated if you knock out PKD and do nephrectomy. So the gene expression is increased. We don't have a good antibody for the actual protein, but we do have an antibody for those V0, V1 neoepitopes that I was talking about. It cleaves the V0 and V1 forms of Versican. And this is the quantification. This is the Western blot, and down here's the IF. And what you can see is that the cleavage of Versican and generation of those neoepitopes is increased with hyperfiltration in normal kidneys and further in PKD. And it's right in the tubular basement membrane. This is the PKD with nephrectomy here. And you can see how strongly cleaved Versican is in the basement membrane with nephrectomy. You can see it a little bit over here in wild type kidneys with nephrectomy, but more so in PKD and PKD nephrectomy. So we've knocked it out. I'm almost done, don't worry. We've knocked it out. And when we knock it out, we see a reduction in the kidney weight to body weight. This is what the kidneys look like at 18 weeks. Morphologically, they look better. Cyst index is improved. So it does appear that this is important for the physiologic and pathologic remodeling of the tubular basement membrane that is being regulated by the macrophages, we think, and that this type of understanding of how the TBM is remodeling might give us new ways to approach therapy for cyst expansion in PKD. I want to credit the people in the lab who've really done the work. Vijay Kakati, Zafer Ackman are actively working on this right now and working on these papers. Lei-Wan Zhu has helped with us, as many other collaborators in my funding. Thank you.
All right. Thank you, Lloyd. I think we already got some questions. So the first one is like, does MCP1 have direct effects on epithelial cells independent of immune cell infiltration?
Does it have what type of effect? Sorry.
Does MCP1 have direct effects?
Oh, direct effects. Oh, yeah, sorry. We don't know that MCP1 signals to the tubular cell. The tubular cell is making MCP1, but other cells can make it. Once the macrophages are recruited, the macrophages themselves make MCP1, and Lei-Wan Zhu has shown that they essentially become a nidus for persistent recruitment of new macrophages, because existing macrophages, once they're activated into the late profibrotic phase, will make MCP1 and recruit other macrophages. I don't know of data showing that MCP1 directly affects the tubular cell itself.
The next one is like, was there an effect of NX on the proliferative activity, example like Ka67 marker, matching the effect on cystic development?
Yes, so that's all being done by Zafer Ackman right now. There's a very clear burst of proliferative response after nephrectomy in a wild type mouse, as well as in a PKD mouse, and it's accelerated in PKD, as you might expect, compared to wild type. So, but even in the wild type mouse, there is, so again, this is why I think physiologically, everything we think about in PKD is actually a normal physiologic response to an increase in GFR that's just dysregulated if you don't have the brakes. So the cells proliferate, they hypertrophy, the tubule basement membrane remodels, the tubule gets bigger, but it's supposed to turn itself off when the GFR is back into the right place and the morphology is correct. And the turnoff doesn't happen if you don't have the polysystins. So I think that it's a dysregulated physiologic process. That's how we're trying to think about it.
Is Versican cleavage related to increased permeability and inflammation?
Yeah, so permeability and inflammation. So I didn't present, so that V1, one of those two epitopes, the 70 kilodalton fragment that's released when you cleave V0 and V1, has another name, and that name is versicine. And it's because it's been shown in different systems, not in the kidney and not in this area, as a pro-inflammatory macrophage activator. And so we actually think that the excess cleavage of Versican by ADAMTS1 and PKD is activating the pro-inflammatory signal in the macrophages by this Versicine effect. Now, permeability, we haven't studied. I can't directly answer that part, but inflammation, yes, we're pretty convinced about that. I didn't have time to put that in for today.
Okay. In the UNX model, why would tubule dilation necessarily increase tubular transport?
Oh, it doesn't increase the tubular transport. I don't mean that. It's the tubule if you need to transport 10 times more solute, then you need more mitochondria, you need more transporters available, you need more brush border area. I think it's just matching the tubule size, tubule morphology, and lumen size to that increased GFR.
Okay, and when you refer to hyperfiltration, did you see a difference between filtration rates along the length of the tubule, or was this outside what you could define?
Yeah, this is another thing that Zafer's taken on, which is I only showed you S1 segment, but as you all know, there's many segments in the nephron, and people are more interested actually in the collecting duct for PKD than they are in the proximal tubule. We've now improved the technology to the point where we can get pretty reliable data on S1, thick ascending limb just before the macula densa, DCT densa, and collecting duct. And so we're going to have that data. It's throughout the tubule. The short answer is the whole nephron remodels in response to hyperfiltration. But I don't want to answer it too specifically yet, because in PKD, those differences there may be real differences in how each segment responds to hyperfiltration and PKD. Don't have that answer yet.
Okay. I think with that, that's it for the questions. And thank you, Lloyd, once again. And we can
thank you for inviting me. It was fun.
We have one more question, actually, and I think you have time to answer it. Sean asked first, regarding to the tubule remodeling, have you checked any other acute injury model other than nephrectomy? And do you observe cell death during the tubule remodeling process?
Yeah, so we've done this in ischemia reflow, but not ischemia reflow in PKD. So I didn't present any of that data. And one of the reasons we did, that's a great question. One of the reasons we thought about that is because the pathologists are always talking about tubule dilation in human biopsies with ischemia reperfusion or hypotensive kidney injury. And what we found in the mouse model is there is no change in tubular diameter in ischemia reperfusion injury. The cyst, what they call the tubule dilation or whatever, is basically because there's so much death of the cells that the lumen gets huge, but the actual tubule doesn't change in morphology. We published that in AJP about a year ago, I think. That's the only model we've checked.
All right, thank you.
All right, thank you.
Oh, so the next speaker, go ahead.
I have the pleasure of introducing our second speaker, Dr. Sorin Fidelis. He is the Executive Director of the Polycystic Kidney Disease Outcome Consortium at the Critical Path Institute and an assistant professor at Yale School of Medicine. His laboratory focuses on target identification and validation for polycystic kidney and liver diseases using whole genome forward screens and pharmacological approaches. The title of his talk today is Modulation of Hypomorphic Polycystin 1 Species in Vivo Ameliorates ADPKD Progression. Please, Dr. Fidelis, take it away.
Thank you, Amanda. Can you hear me?
Yes, we can.
Perfect. Let me see if I can share the slides. All right, here we go. So I'd like to thank the organizers for the invitation to present today. These are my disclosures. So, we've heard from the wonderful speakers about PKD pathophysiology, so I will not spend a long time going over that. I will talk to you about the genetic and molecular connection between isolated polycystic liver disease and the unfolded protein response, and how those studies led to the current story that we published this year on a new hypomorphic mouse model for ADPKD, and how we have used that to examine approaches to modulate PC1 folding/function. All right, so I'm not going to go through this slide. As you know, PKD still remains an area of huge unmet need. One drug approved, a couple of phase 3 trial failures in the last year. Lots to do, lots of new targets and better drugs needed. In terms of the pathways, a lot of pathways have been implicated in PKD that are downstream of the initial genetic lesion. And while this approach has yielded interesting results, and the therapeutic approach and the approved drug that we have right now, we are clearly focused on that initial genetic lesion and on ways to modulate that process by using as a target the polycystin-1 protein itself. Hard to work with PC1, huge protein, lots of interesting domains. A lot of interesting work has been done, including the structural crystallography work that elucidated the structure of the heterotetrameric channel. Again, the protein is processed many different ways. You've got the GPS cleavage. Obviously, you've got other cleavages in the C-terminal region. And, this presents opportunities to obviously target some of these processes in terms of therapeutic approaches, but also challenges because of the complexity of, processes, mechanisms that are at play in this context. In terms of the genetic mechanism, I'm not going to focus the talk on mechanisms themselves, but suffice it to say that people have been working on this for the last 25 years. You've got the two-hit mechanism, or you've got a germline mutation, somatic second hit. You have this slow growth over decades, but also approaches that people have looked at whereby this dosage type mechanism has been characterized by many people in the space. And what this entails really, let me put the laser right here. So what this entails is that, in a polycystin-1 dosage and time-dependent fashion, you can have faster or slower cyst growth depending on where you are along this polycystin-1 continuum. So obviously, from a therapeutic standpoint, this has clear implications for cases where, you've got polycystin-1 species that still maintain function, that are along this continuum whereby interventions that increase the levels of polycystin-1 species/function can bring you into a different category of cyst growth. In other words, slowing down cyst growth progression. This is taken from Greg's talk at the RC Symposium a couple of years ago with his permission. But I thought this illustrates really nicely how we think about the dosage mechanism. Obviously, we all, or most people, they walk around and they have one somatic hit in polycystin-1, and they're perfectly fine. And, there's no cysts taking place in this context. Now, when you have a germline mutation in PKD1 or PKD2, and then you combine it with a somatic second hit, where that hit lies along this PKD1 activity continuum can either mean that you maintain function above a certain threshold and no cysts develop, or you get this accelerated cyst growth that occurs due to falling below this threshold. And as you can appreciate, you have nuances to this, right? It's not all or nothing. You can fall along this continuum, or you've got enough function to be above the level required to prevent cyst formation. You can be a little bit below. So thinking about ways to push this PKD1 activity/levels along this dosage interval, can potentially make a huge difference in terms of disease severity and progression. And why do we think about this, and where this all started, at least for us, was with the isolated polycystic liver disease phenotypes, where you have this ER proteins, whereby mutations in genes that encode for this ER proteins involved in co-translational protein translocation give you this cystic phenotype. Initially, PRKCSH63 were identified as the isolated polycystic liver disease. genes whereby you have this liver phenotype that phenocopies the one seen due to PKD1 or PKD2 mutation. In the last few years, a plethora of other genes in the same really pathway of co-translational protein translocational folding have been identified. And as you can appreciate, you've got GANAB, which essentially is the same protein, the alpha subunit, catalytic subunit of glucosidase 2, which complexes with glucosidase 2 beta. And then you've got other proteins involved in this pathway, 61 beta involved in the translocation process. And then you've got, ALG8, DNAJB11, LGA9. And where they all act is this pathway where you've got this nascent polypeptides that are threaded through the ER translocon. And then you've got these processes that generate this glycan that gets attached to the nascent protein. So you've got ALG9 as part of this pathway. Then you've got downstream of glycan attachment to the protein. You've got this proteins involving glycan processing, such as glucosidase 2 alpha and beta. And as you can appreciate, this process is a housekeeping mechanism, right? But proteins that are very large, like polycystin 1, will be very sensitive to disturbances in this pathway. And this is exactly what happens in the context of disturbances in proteins in this pathway. So we, many years ago, too many really, showed in vivo that this disturbances in this polycystic liver disease genes really impinge upon polycystin 1 dosage. So in this case, when you inactivate PRKCSH, so that's the beta subunit of glucosidase 2, you get bile cyst formation. When you decrease PC1 levels to half on the same background, you get a lot more severe cystic disease. When you do the same with PKD2, it's an intermediate effect. And when you do the opposite, when you increase polycystin 1 by 3 copies over baseline in the context of PRKCSH deletion, you pretty much rescue the phenotype at this time point. If you wait longer, cysts do form. And then if you do the same for polycystin 2 overexpression, or mild overexpression, if you will, no rescue is seen here. So we concluded based on this study that polycystin 1 really is the master regulator of the cystic phenotype due to inactivation of PRKCSH and 63. And really, this is the key to this pathway. Okay, this pathway impacts polycystin 1, and this was shown really clearly in vivo through the studies. Interestingly, if you modulate PC1 levels via pharmacological approaches, in this case, you inactivate the proteasome and you can increase polycystin 1 levels, seen here by an increase in the CTF levels, compared with the PRKCSH mutant cells that weren't treated with NG132. And then you do the same in vivo, you do see an amelioration of the phenotype that, presumably goes through an increase in polycystin 1 levels. So this was an indication that, again, modulating PC1 levels in this context of, glucosidase 2 beta inactivation can have an impact in vivo. Based on these studies, we looked at the next question, which was quite obvious. What happens when the unfolded protein response, which again plays into disturbances in ER proteostatic mechanisms that are driven by these PLD effectors. What happens with respect to UPR impinging on cystic disease progression. So we looked at UPR pathways that are regulated in the presence of unfolded proteins. And we found that the IRE1 pathway, which is the most conserved from eastern mammals, really does play a big role in cyst progression in the context of Sec63 mutations or inactivating backgrounds. So here, shown on P21, if you inactivate Sec63 in the kidney, this is the early model in the using a KSP-3, you get moderate cyst formation. You do the same or you inactivate 63 and XBP, which is the main pro-chaperone effector of the unfolded protein response. You get genetic interaction, you get a more severe disease. If you do the same experiment as in the previous study, where you increase polycystin-1 levels with the PKD1 back transgene, you partly rescue the phenotype, whereas with the PKD2 back, you don't see an effect. And if you sort of go along this mechanism and investigate whether XBP can play a role in vivo in the context of a PRKCSH knockout background, it was interesting to see that if you increase XBP in cells that are lacking PRKCSH, you get an increase in polycystin-1 steady-state levels in terms of CTF at full length. If you do the same with an XBP ROSA-FLOX stop transgene, where XBP is turned on in a pre-dependent fashion, exactly in the segments that are lacking PRKCSH, as you can appreciate here, we see quite a large rescue of the phenotype when XBP is turned on in the PRKCSH knockout tubules. So based on these studies, we wonder whether this effect of XBP via its pro-chaperone function can apply to contexts where, the folding environment in the cell is normal, but the actual folding of PC1 due to pathogenic missense mutation is present. So in this case, we chose the R2220W mutation, which occurred in trans with the RC mutation in an early onset PKD family. And we looked at this mutation both in vitro and in vivo and in the context of XBP1 upregulation. In cells, if you express this mutant, the RW mutant, you see a cleavage defect, not very clear here in X cells overexpressing the protein. You'll see it better in cells isolated from the kidneys. But anyways, you get a defect in polycystin-1. If you increase XBP in the same cells that are expressing this PC1 mutant, you see an increase in steady-state levels. So this mutation, sorry, I forgot to mention, occurs in the REJ domain. So based on this, we decided to build the mouse model that corresponds to the human mutation. This is the R2216W model. The homozygous mice at P11 are already quite cystic, P16 very cystic. They do have a liver cystic phenotype as well. And overall, they display a very moderate to severe cystic phenotype in the homozygous configuration. So we use this model to essentially investigate whether XBP in vivo can modulate disease progression. As you can appreciate, we can detect the mutant in vivo using the knock-in V5 tag at the C-terminus. High expression in the lung is expected. And then if you look at the ratio between CTF and full length, this is by lysate and this is by IP, the ratio is switched from mostly CTF to a little bit of full length in the wild type to about half and half CTF and full length, which suggests a cleavage defect, which recapitulates what was previously shown in the field. So if you do the experiment in the mice where we express XBP again in the segments that are expressing the RW mutant, we see an interesting phenotype. So what we did here was to create an RW flux line. So we inactivate one copy of PC1, and then we have only the RW present. This is the PKHD1 Cree early model. So, we are doing this only in the collecting duct. As you can see, at P16, we see quite a severe phenotype for the RW flux using this neonatal model. If you express XBP at the same time with flux inactivation, you see a significant, rescue in disease severity in terms of kidney body weight, cystic index, and kidney function. Importantly, if you do the same in mice, exactly the same mice, but these mice do not express RW. So they are PKD flux, there's no protein. You do the same experiment with XBP, expressing it in the collecting duct via the PKHD1 transgene, Cree, you see no effect. So this obviously was important to do. It's gratifying because it shows that the effect that we see is RW specific. In terms of contribution of apoptosis proliferation, we saw a clear decrease in proliferation in the mice expressing XBP. with no significant impact on apoptosis. So this suggests that activation of XPP drives this rescue in the phenotype via decrease in proliferation. In terms of the biochemical effect of XPP, what we did was to isolate microdissect tubules from this RW mice, either with and without XPP. As you can appreciate, the mice where XPP was turned on do express XPP, as shown by Western and IF, and also via the canonical XPP target gene ERDJ4. If you look at the effect of XPP on PC1, again in these cells dissected from the mice, the effect is quite clear. So you have the wild type here, where you have mostly CTF, a little bit of full length. The RW mutant species, which expresses much less CTF, a lot more full length. So, signifying a cleavage defect. When you express XPP in the cells, you partly normalize the phenotype. So you increase CTF while also increasing full length. So if you quantify the ratios of CTF and full length, so you do have an increase in CTF, you do have an increase in full length as well, whereas the ratio does seem to be different. But again, based on this experiment, we cannot conclude that XPP does anything to promote PC1 cleavage itself. It does increase both CTF and full length, and we still need to determine whether cleavage itself is impacted. But nevertheless, this effect is also seen via an increase in ciliary trafficking. So in wild-type cells, you can detect PC1 cilia no problem via the V5 tag. In the RW knockout, it's barely detectable in cilia on a few cells. If you express XPP, you see expression in the cilia, not quite at the level of wild type, but greatly normalized. If you do the same experiment in adult mice, where you knock out PC1 flock, the one copy, you remain only with the RW species. You do this at 6 weeks, you wait until 18 weeks, you have mice that are quite severe. If you do the same, but you express XPP at the same time, you see a partial rescue in the phenotype. Interestingly, if you use a chemical chaperone in the cells isolated from the mouse, it's quite gratifying to see that we can partially rescue a PC1 cellular trafficking in this RW cells using a chemical chaperone. And if you look biochemically in the 4-PBA treated RW cells shown here, the mutant cells have a clear cleavage defect, as shown before. In the 4-PBA treated cells, you do have a partial normalization of the phenotype in terms of increasing CTF and somewhat of a decrease in full length. So if you quantify this, you do have a trend towards a decrease in full length with 4-PBA in the RW cells. You do have a clear increase in CTF, and if you look at the ratio, you have a partial normalization of the ratio. So, we are very excited about this model. I think it's a really good model to study polycystin-1 hypomorphic species, polycystin-1, trafficking function, etc. So currently using this model to test other chemical chaperones and, to really do the ultimate experiment, which is the in vivo experiment for this, chemical chaperone therapeutic experiment. So in terms of where does this lead us in terms of how do we use these models to apply them in a therapeutic? Yeah, 30 seconds. This is my last slide. To apply this in a therapeutic setting, this allows a model where we can study polycystin-1 trafficking, allows for screening of chemical chaperones or performance screens where we have as a readout, PC1 cellular trafficking as a primary phenotype, and then potential channel function as a secondary screen. So in conclusion, through these studies, I hope I have convinced you that XPP1 can promote the steady state levels and processing of PC1, that the RW model is a good model to study these processes, and that potential activation of XPP1 dependent chaperone pathways can have therapeutic utility for a subset of PC1 missense mutations. yeah, I would like to thank Rishi Bhardwaj, very talented postdoc who performed a lot of the experiments, Isabel Volpe, and my collaborators, Steve Samlo, Mateusz Krapitz, who was a co-first author on this JSON study, Anna Rachel Gallagher, and Whitney Bessie, and everybody else who was involved in this research, and take any questions.
Wonderful talk. Thank you so much. So our question in the chat says, do you think 4-PBA is having an impact more as an HDAC inhibitor altering proteostasis rather than a chemical chaperone?
So somebody works with PBA. I can see that. That's great. Obviously HDACs, there is evidence that HDAC modulation may play a role in PKD. We are examining that question right now. So essentially see whether the 4-PBA effect, if in the case of going through HDAC, should be applicable to backgrounds of complete PC1 loss of function, so like a PC1 flux model. Whereas if it went only through the RW modulation, it should only apply to RW background. So that's exactly what we're trying to answer, both through in vitro and also through our in vivo models. Great question.
Thank you. I'll ask a quick question while we wait for anyone else that has questions. What do you think, or do you think these mechanisms play a role in ARPKD, seeing as there is a multi-tissue effect, as you showed the liver and kidney disease effects here?
Can you repeat the last part of the question? Sorry.
Just in the context of it having liver and kidney effects, how do you think this plays a role, or the proteostasis?
Right. The liver sensitivity versus kidney sensitivity to polycystin-1 dosage and where you are along this continuum. I mean that's the question everybody asks. Like, why do you get a liver phenotype and not a kidney phenotype in people sometimes? And then, again, we're trying to understand the specificity of that sensitivity to PC1 dosage. We haven't, in the context of the RW model, which has a liver phenotype as well, as I mentioned, we haven't done that experiment where we express the XBP1 transgene in a Cre-dependent fashion yet. That's being performed right now as well. So I can't answer the question regarding the effect of XBP increase / chaperone increase in the context of the PLD phenotype, but we're doing that right now.
Okay. So does the RWPC1 protein traffic to the membrane?
So we have not looked at the membrane trafficking as a readout for our studies in terms of polycystin-1 trafficking / maturation. So I cannot answer the question, but that would be great to know, because again, that can be utilized for certain readouts. So that's a great question. And
what was tubule identity of remaining cyst in the RW Flox XBP model?
Right. So the tubule identity in the in the PKHD1 Cre RW Flox model with XBP with or without, those were the remaining cysts were collecting duct origin. In terms of the PAX8 model, the differences in cystic burden in the partially rescued animals, I don't have a clear answer for that. The effect was seen both in proximal and distal phalangic duct regions in terms of lower disease burden with XBP expression, but I don't have a clear quantitation for that to tell you exactly. Thanks for the question, Robin.
For PBA, it was used for correcting CFTR. Did you look in the model of CFTR expression? Was it increased?
We did not, and thanks for the question, Ludmila. And yeah, I think that's a very interesting well, not readout, but very interesting, target to look at and to see the effect for PBA in our RW setting. And we'll make sure to do that. Thanks for the question.
And our last question, any thought on how the RAJ mutation affects cleavage and ciliary trafficking of PC1?
Fung. Short answer, no. It's we have
time.
Yeah, sorry. We have we think that the XVP effect that we see is domain specific. So we have looked at other mutations with XVP upregulation, and we, this was not in the paper, but we the effect that we saw seemed to be specific to the REJ gain domain in terms of the impact on PC1 steady state levels and cleavage. But I do not know the exact mechanism, and I don't want to speculate right now. But thanks for the question.
Thank you, Dr. Fidelis.
All right, let's move on. Our next presentation is an abstract presentation by Christian Charbonneau from Johns Hopkins University School of Medicine. The title of his presentation is like a CFTR-based therapy for autosomal recessive polycystic kidney disease. Please proceed, Christian.
Hello, I'm Christian, a postdoctoral fellow at Johns Hopkins. I would like to thank the organizers for giving me the opportunity to be here today. I'll be presenting our work on CFTR-based therapy for autosomal recessive polycystic kidney disease. It has been postulated that CFTR inhibitors could be used for PKD treatment, but Abner's group mixed their PKD with CFTR knockout mice, and we've seen that the disease got worse. And recently, PKD and CFTR knockout mice didn't show a stop in the disease progression. Thus, CFTR inhibitors could not work. On the other hand, CFTR correctors, they move the CFTR from the ER, where about 80% is stored, and move it up to the membrane. And it was reported that in AD-PKD we have well-differentiated isolated cysts, and this is what we've seen in our CRC model. And on treatment with the VX-809, which is a CFTR corrector, we've seen a significant reduction in cysts. And in AR-PKD, the cysts are fusiform, and in about 10% of our DEL3-4 mice, we've seen a significantly increased size in the kidneys. But in the liver, we have multiple cysts present that respond well to treatment with the VX-809. In the early stage, we took 5-day-old mice and treated them for 30 days with VX-809, which significantly reduced the dilatations in the bile ducts. It decreased the proliferation, and while in AR-PKD, we find more CFTR on the apical membrane, and the treatment with VX-809, we find more in the basolateral membrane. We tried to look at the difference between the early stage and later stages. So we took these 5-day-old mice and treated them for 5 months. And we also took a group of 4 months and treated them for 1 month. And we observed a significant decrease in the cyst area and an improvement in the liver function. The CFTR protein expression was increased after treatment, and the proliferation was decreased in both cases in the early stage and late stage. The in vitro studies with the cholangiocytes that were isolated from DEL4 mice, we've seen that in the treatment with VX-809, the cysts have significantly decreased, while in the treatment with the inhibitor 172, which would inhibit the CFTR, the decrease in size was less significant. And looking at the localization of CFTR in the cysts and in the cells, we've seen that there's more CFTR at the basolateral membrane after the treatment with BX809. Cyclic AMP and intracellular calcium release is high in ARPD. And with treatment with BX809, we were able to decrease both the cyclic AMP levels and the calcium release. And the protein's level of CFTR was increased after the treatment with BX809. The heat shock proteins, which are involved in trafficking and degradation of the proteins, are highly upregulated in ARPD and have been decreased at HSP7, HSP90, and AHA1 in the treatment with BX809. In order to show the direct link between CFTR and ARPD, we resulted in gene therapy. So we used a truncated form of CFTR made by Dr. Sputaro, where the first 4 loops are missing, and this was able to be packaged in AAV1. And in cells, we see an increase in chloride function. And treating mice with this AAV1 delta 24 to 64 CFTR with 1 dose at 1 month and sacrifice 2 months later, we see a significant decrease in the cyst area. The expression of CFTR increased and the proliferation has decreased. So BX809 reduces the cyst area in the cells. It reduces dilatations of cholangiocytes and proliferation in the liver tissue. The CFTR expression is significantly decreased at the apical membrane and increased in the basolateral membrane of the cholangiocytes after treatment with BX809. And the treatment 1 dose of AAV1, delta 27 to 64 CFTR, we've seen a decrease in cyst area and decrease in proliferation. So in our future work, we'd like to understand the role of CFTR and the mechanism of ARPD and potential gene therapy for ARPD in the truncated form of CFTR packaged in the AAV1. I'd like to thank Dr. Sputaro for her continuous support and guidance, Dr. Murali for laying the strong foundation of this project, Dr. Peter Harris for providing the human and cells and tissue, and Dr. Salmo for providing the mouse cells, Dr. Patricia for the support of the project, and the Kidney Center, University of Maryland. And I'd like to mention that all this is possible with the support of the NIH and the Kidney Foundation. Thank you.
Okay, well, we wait for the question. I'll start with the question. How did you deliver the AAV virus, and what is the distribution across the kidney?
So we delivered the virus via intraperitoneal injections. It got more distributed in the liver tissue, and we have seen an increase in CFTR in the cholangiocytes, but it was more in the liver tissue compared to the cholangiocytes.
Any other questions?
Yes, we have one question in the chat. Does the effect of the VX corrector of liver cyst on PKHD1 protein, is it on the PKHD1 protein rather than on CFTR?
So this is what we wanted to show with the by using CFTR in packaged in AAV1. We wanted to see if it was the BX809, if it influenced the PKHD1 or the heat shock proteins, which we know they do influence, or do we have a more of an effect from CFTR itself by increasing the CFTR, and that causes the cyst to reduce. And our preliminary data showed that it might be that CFTR alone was the cause of this. But yeah, we're looking into it.
So the next question is, like, could you try on a PKHD1 null model?
We
Have you
don't
tried that thing?
we don't have a PhD1 null model at the moment, but yeah, that's a good thing to look into. Thank you.
All right. If there are
Okay,
any more...
I'm sorry. We're gonna have to stop and move on. Can we move on to the next presenter, please?
Sure.
Thank you, Christian.
Thank you.
Our next abstract presentation is by Madeline Gaithier. She is presenting on the role of renal nerves in a preclinical model of advanced autosomal recessive polycystic kidney disease.
Okay. So thank you for that introduction. My name is Madeline. I'm a PhD candidate at the University of Arizona in Dr. Christopher Bandick's lab. And as she said, I will be talking to you guys about the role of the renal nerves in polycystic kidney disease. So as we're all well aware, treatment options for polycystic kidney disease are extremely limited. Patients who enter kidney failure can either undergo hemodialysis or can wait for a kidney transplant, but neither of these options are ideal. Tolvaptan, which is a V2 antagonist, acts on the arginine vasopressin pathway by inhibiting the V2 receptor. And this is effective because AVP is increased in polycystic kidney disease. However, tolvaptan comes with the significant side effects of polyuria and polydipsia. Now we know that AVP is increased in PKD, but our lab has chosen to approach this component of the disease from a somewhat novel perspective, that of the afferent or sensory renal nerves, which project from the kidney to the brain. Now, there's limited but compelling evidence that these nerves may contribute to the release of AVP. And this comes from a series of studies originally done in the 80s, but we also have preliminary data that supports this from our laboratory. And some of the evidence and data from one of those original studies is shown here in this graph on the right. And what the group did was they stimulated the afferent renal nerves, and then they collected blood samples before and after. And what they saw was that plasma AVP levels were elevated at 5 minutes post-stimulation compared to baseline, suggesting that these renal nerves were contributing to AVP release. Now, the concept that afferent renal nerves may be contributing to a cardiorenal pathology is not novel. Afferent renal nerve ablation attenuates hypertension in the Doka-Salt model of hypertension, and there's increased afferent renal nerve activity in this model. Furthermore, the afferent nerve activity is associated with inflammation, which may be driving this nerve activity. There's also substantial overlap between polycystic kidney disease and hypertension, so it's not too big of a stretch to suspect that there may be a role for these afferent renal nerves in polycystic kidney disease. In fact, last year, our lab published a paper that showed that in PCK rats, there was significantly greater afferent renal nerve activity compared to age-matched healthy Sprague-Dawley controls. And when we took that afferent renal nerve activity and we correlated it with the animal cystic index, we saw a significant positive correlation. And then finally, when we removed those nerves through either total renal denervation or an afferent renal nerve ablation, we saw a significant decrease in cystic index compared to sham-treated controls. Now, the data that I showed you on the last slide were from studies done in younger animals from 4 to 10 weeks of age, but we wanted to examine the role of renal nerves in animals who were 24 weeks old or 6 months old, because these animals have significantly more advanced disease. And because the disease is more advanced, we assumed that cystogenesis would be so progressed that we wouldn't be able to affect it with our intervention, but we thought that perhaps we could still attenuate the decline in renal function or decrease blood pressure. Thus, we hypothesized that neither total denervation nor afferent denervation would decrease cystic index, but that total denervation would drop systolic blood pressure and would attenuate the decline in renal function. And to test this, we used male and female PCK rats who were 24 weeks old, and we performed baseline measurements and then performed the treatment surgeries when the animals were 24 weeks of age. We then followed them out for 6 weeks, repeated those measurements, and then euthanized and collected tissues. And throughout the study, we measured blood pressure through tail cuff. And these are our results. So on the far left graph, you can see the renal cystic index. And we saw that in contrast to our original hypothesis, we were able to decrease cystic index in both total denervation and afferent denervation animals, suggesting that afferent renal nerves were contributing to cyst growth in this model, even in this advanced disease stage. As I mentioned, we also measured blood pressure via tail cuff, and we saw that the total denervation group, as shown in the empty triangles, had a significant decrease in blood pressure relative to sham-treated controls, but this was not seen in afferent renal denervated animals. We also measured GFR via the transdermal measurement of Fitzy-Sinistron clearance and saw a significant drop in shams GFR, but not the other groups, suggesting that we attenuated that decline in renal function. And finally, we measured serum copeptin, an indirect indicator for vasopressin, but failed to achieve significance in either treatment group, suggesting that perhaps we were not affecting the vasopressin pathway. So to conclude, I've shown you that both afferent and total renal denervation decreased cystic index, suggesting that afferent renal nerves were contributing to cysts in this animal model. And I also showed you that total denervation decreased blood pressure, and both treatments attenuated the decline in renal function. However, these results leave many questions unanswered, namely the underlying mechanism by which we're affecting cystic index. The overarching hypothesis with which we're working right now is shown in this figure on the right. So we hypothesize that the damage induced by early stage cystogenesis, such as inflammation and fibrosis, is driving up the activity of the afferent renal nerves, which increases the secretion of vasopressin, which acts on V2 receptors to increase cyst growth, causing a vicious cycle. However, another outstanding question is how we are affecting cystic index since these animals have more advanced disease. Are we simply slowing the progression of additional cysts and the cysts that are already present, or are we actually reducing the size of the cysts that are present in the kidneys prior to treatment surgeries? So in future studies, we'd like to address these questions and many more. But to conclude, I'd like to acknowledge the many members of the Bannock Laboratory, as well as our funding sources. And with that, I can take any questions.
Thank you so much, Madeline. Unfortunately, we are over time, so if you have questions, please add them to the chat and we can address them there if that's okay with you.
Of course.
All right, our next presentation is a cells model subcommittee presentation by Professor Paul Willing. Paul, please proceed.
Can you see the screen?
Yeah, looks good.
Wonderful. Thank you very much. This is a presentation about the cell models offerings from the PKD RRC. I am chair of the committee. John, Owen, and Darren are the site leaders, and the various committee members from all of the centers. The cell models group provides a number of reagents for the community, including human epithelial cells, both healthy and from ADPKD cysts, immortalized cell lines that allow the conditional inactivation of polycystin 1 or 2, primary mouse cells from animals bearing conditional alleles for the polycystins, newly developed iPS cells that have inactivated polycystins, and we're developing cell lines, human cell lines with the ability to conditionally express the polycystins. One cell line in the HEX cell model has been developed. The renal tubule epithelial cells are in development. I'd like to spend most of the remainder just talking about new developments and reagents and offerings. I'd like to say before I get to that, I'd like to say that all of the reagents that are currently available have gone through extensive quality control and protocol development. They're available through a web-based ordering. We also offer a point of contact for consultations. You can download the PDFs that show you how to grow the cells, what to expect from the cells, and kind of the stage of development and QC analysis. So for the last 4 slides, I'm going to just tell you about new developments and areas that we're currently working on and new offerings. We're very happy to report that the first polycystin 2 inducible mouse knockout has been made. This was made and extensively characterized and is now available to the community. This was
2 minutes.
based on the Paxate TET inducible system and crossed on a Morta mouse. You can see on the left all the steps that we've characterized, including RNA-seq profiling before and after the addition of doxycycline, and immediately after the knockout of polycystin 2. Unlike previous presentation, the transcriptome stays relatively constant in passage, and the knockout of PC2 along passages induces comparable changes along passage. This data is available to the community. Please ask us for this. We're in the process of easy web-based acquisition. We also have developed PC1 knockouts from both cortical tissue. These are in various stages of development and characterization. They are able to acidify the media upon knockout of polycystin. They grow cysts in matrix. We also have a medullary clone that has a very interesting phenotype. It seems to grow more tubuloids in matrigel and then obvious cyst formation upon knockout of the polycystins. At Alabama, they're developing the iPS cells. Available are knockouts of polycystin 1, 2, the heterozygote PK. Yep, and some that are under development that allow one to study cystogenesis in this model. And here are some examples from some recent publications of what can be done. So I'm out of time and we'll leave this for discussion and questions. Thank you. It sounds a bit like
looks like there's one question in the chat.
Okay, yeah. Is it possible to send the PKD 1 and 2 human iPS cells to Japan? Do you also have the control iPS cells?
So we have been able to ship frozen cells globally. This takes a little bit of work. We're willing to help you do that as long as you pay for it, the shipping and all of that, and you make the arrangements for customs and all of this. This is really the hard part, is getting the paperwork done. And in our experience, really the recipients need to be responsible, and they're the best ones informed how to deal with all the customs issues. But we have successfully shipped them around the world, and we're happy to try Japan. I didn't get the second part of the question.
Do you have any like control iPS cells? Like maybe the isogenic controls?
So they're not strictly isogenic, but they're from the same clone, and those should also be available, sure.
And what cell models are available for ARPKD studies?
So they are being developed in the iPS model. You saw one there listing. That's where we are with that.
Okay. And we can have one more question that is like,
I said no.
okay, immortalized cortical PKD1 cells are LTL positive proximal tubule in nature?
So those are proximal in nature. The goal of the mouse lines is to generate male and female from each of the major segments of the nephron. And so that's kind of the approach, by going cortical, hoping that we get mostly proximal, going medullary, hoping more for at least the medullary collecting ducts. We may have to do some pre-sorting to identify the other segments of the nephron, and we're in the process of thinking about how to do that.
Thank
right.
you. Unfortunately, we're going to have to stop. So I'm going to send it back to Brad.
And I want to kind of close this out, thanking, first of all the speakers and moderators for a wonderful job today. You know, I apologize for cutting some of the conversation short here, but if you do have questions, you know, concerns or anything like that, or resources you want generated or considered to be generated, please do not hesitate to reach out to us in the consortium, and we will do what we can to get these resources generated. I also want to again thank CJ and Patricia for organizing this whole symposium. It's a lot of work that went into that, as well as the committee. Terry, did you have anything that you wanted to add before we head this off to the trainees symposium?
No, I just want to thank everybody. It's really great to have so many people online, and I'm glad that everybody participated, asked questions. The speakers were great. Moderators were great. It was a great 2 days. And so just people who are going to the trainee session, the link was sent out. If you, for some reason, didn't get it and you're a trainee and you want to join, please email myself or Nicole and we'll share it.
I do have one last thing. I did want to send out a big thank you to Nicole for taking care of dealing with a lot of the administrative stuff behind the scenes. So she was right there behind, sometimes, shoving Katrina and I along.
Yeah, ditto. Nicole, great.
Thank you. Thank you.
Thank you, Nicole.
And a great timer. I already got a comment from someone who said, I want that timer.
I love the timer.
Thank you. Glad to help.
Just as a final reminder, we will be sending a survey out. Please do your best to fill that out. We value your input on how we can do things better. So thank you, everybody, and I hope you have a great rest of your week. And the trainees, have fun in your session.